For today's revision class on the chapter Human Health and Diseases, in which today we will discuss the most interesting and important topic of this chapter, humoral and cell mediated immunity. About this topic, I have already given you the notes in the earlier class. Now, today you will try to note down some of the important points related with these two aspects of our immunity. Now, we will start with the topics which are we covering in this antigen, cell mediated immunity, humoral immunity, antigen antibody complex, and mechanism of action of antibodies. Now, first of all, what are antigens? The word antigen is derived from the term antibody generating. An antigen refers to any substance that can evoke an immune response. Foreign proteins triggers the production of antibodies. So, proteins are known as complete antigens. Lipids and carbohydrates can trigger antibody production in combination with proteins. Such antigens are called haptens. Antigens are usually present on cell walls of microbes and cell membranes of body cells, either host or foreign. The specific region of an antigen that binds with an antibody is called an epitope or antigenic determinant. Now here you can see the self-antigen, the major histocompatibility of the complex, how it is going on. As we know that all cells, including our own body cells, have glycoproteins and lipopolysaccharides on cell membrane or cell walls in case of microbes. Then how does the immune system differentiate between self and non-self antigens? All vertebrates express a special class of proteins called the major histocompatibility complex, which in short we can say that MHC, on cell surface, and this complex is also known as self-antigen. A self-antigen is any chemical substance in an organism that acts as an antigen on entering another organism, but to which the immune system of the parent organism is tolerant under normal conditions. The MHC is used to determine compatibility between a donor and a recipient before organ transplantation. This is the significance of that MHC, means major histocompatibility complex. Here in this diagram you can see that how the self-antigen of a particular cell, when mismatched kidney cell of donor 1 and it is matched with kidney cell of donor 2, because it is matching, in that case it can donate the kidney to that particular person. That means in case of organ transplantation, that concept or that fact is taken. Now, here is in this slide you can see and understand pathogen specific immune response. An acquired or adaptive immune response is a response to a pathogen that infects the body either accidentally or through vaccination. This type of immunity is pathogen specific immunity, and pathogen specific immunity is basically developed by the lymphocytes. And as we have learned that there are two types of lymphocytes in our body, T lymphocytes and B lymphocytes. Pathogen specific T and B lymphocytes or T and B cells respond to a pathogen differently. T cells generated and activated cell-mediated immunity, which is known as CMI, and B cells generated and activated humoral immunity. And now, what happens in cell-mediated immunity uh, and how these T lymphocytes develop that cell-mediated immunity? There are three types of T lymphocytes or T cells in our body: cytotoxic T cells, or which are known as killer T cells. They migrate to the site of infection and release cell lysing proteins such as cytolysine and perforin, which directly kill the pathogen. Helper T cells, the another type of T cells, they secrete helper factors, which are certain types of proteins which stimulate B cells to produce more antibodies and enhance the activity of killer T cells. And the third category of T lymphocytes are known as suppressor T cells, and they secrete suppressive factors which suppress the T cells mediated response once the immune reaction is over, and they also cause immune tolerance. They protect body cells from an autoimmune reaction. And here is the structure of T lymphocyte or T cells you can see in this image. Now come to the next slide, where you can see how does action of T cell take place during cell mediated immunity. Macrophages process a pathogen and present the antigen on it to helper T cells. Then a series of events leads to activation of cytotoxic or T cells, killer T cells and B cells. Now here in this flow chart you can see that how macrophages respond to generate the cell mediated immunity. Here this is a macrophage cell. Antigen on a pathogen when comes in contact with that macrophage, antigen presentation starts, T helper cells get activated, make activated the B cells, and then activation will start with the help of lymphokines, and T killer cells starts to form, and T suppressor cells are also start to form. Lymphosomal enzymes are released and they cause cell wall lysis or breakdown of the cell wall of that particular antigen or pathogen. In that way it kills that particular antigen or pathogen. The cell mediated immunity is able to select self and non-self recognition. How it will be able to self and non-self recognition? Try to understand this now, and which is also one of the important fact in medical profession or in field of medicine. The cell mediated immune response helps the body to differentiate between self and non-self antigens, and during that CMI is creating an important role, means it is responsible for graft rejection, means this ability of our immune system can reject the graft at the time of organ transplantation. That's why at that time the patient should be given immunosuppressant to suppress its immunity so that graft can be accepted by the body. In case of a mismatched donor, helper T cells identify the antigen present on the cells of the donor tissue as foreign and release lymphokines to activate cytotoxic T cells. The activated cytotoxic cells attack the donor tissue, resulting in transplantation rejection. Similarly, that also create the cancers in case of cancers. That cancer cells develop the cell mediated immunity which also protects the body from its own cells and that have turned cancerous. In that way, our own cells starts to develop under the cancerous condition, or they start to divide irregularly by this ability. That's why cancer is also caused immune disorder also. Now we will talk about humoral immunity and what is the role of B cells in that humoral immunity. When B cells come in contact with an antigen, they divide rapidly to form plasma cells. And plasma cells produce antigen specific proteins called antibodies at the rate of 2000 antibodies per second. The antibodies are released into the blood, they circulate and bind to antigen on circulating pathogens. As the antibodies are present in the body or body fluid, which is known as humor, this type of immune response is known as humoral immune response. And here in this chart, you or in this image, you can see that how the B cells get connected with the antibodies to develop that humoral immune response in our body. Now, how does this humoral immunity develop by the action of B cells with the help of this flow chart? Try to understand. Activated helper T cells release again lymphokines which activate B cells, and the activated B cells differentiate to form antibody producing plasma cells and memory cells. Here this is the macrophage cell. Antigen on a pathogen when come in contact with this macrophage cells in our blood, antigen presentation starts, which with the help of which T helper cells activate the B cells, which starts to differentiate plasma cells and memory cells, and lymphokines used to form T killer cells and T suppressor cells, and in that way lysosomal enzymes starts their cell wall lysis to fight against or to eliminate that pathogen from the body. And here when they activate the B cells, what does these B cells do form doing their action? How does they take place? They differentiate the normal cells as which are in the form of plasma cells as well as memory cells, and starts to form antibodies against that particular pathogen, which fight against that particular pathogen or microbe. Now here we are revising with the help of this slide the antigen antibody complex. How does it form? Entry of an antigen triggers a humoral immune response, inducing the formation of specific antibodies, and this is called antigen immunogenicity. The antigenic determinants or epitopes, here this is the epitope, bind to the corresponding paratopes. Here this is the paratope on antigen binding site, and this particular site of the structure of antibody is known as antibody antigen complex were formed, and that paratopes at antigen binding site when get starts to combine with that epitope, then antibodies starts to form an antigen antibody complex, and the formation of an antigen antibody complex follows a lock and key mechanism here, and the chemical reaction between an antigen and a specific antibody to form an antigen antibody complex is called antigen reactivity, that starts here in case of development of our immunity. Now here you can see that the antigen antibody complex are formed in these three cases. How? Opsonization, an antigen antibody complex binds to a receptor on a macrophage. The macrophage ingests the entire complex and digests it. This is one type of antigen antibody complex reaction is going on here, and in that way they phagocytically digest the microbe. The another type of reaction of that antigen antibody complex? Agglutination. Uh, Pentameric immunoglobulin M, like immunoglobulin M molecules, bind to antigens, forming large antigen antibody complexes, and these complexes filter out into the lymph nodes, are later destroyed by other cells of the immune system. And the third type of reaction that you can see here in this image, complement activation. In this type of reaction of the antigen antibody complex, the formation of an antigen antibody complex triggers the binding of a special class of plasma proteins called complements to the surface of the pathogen, and the complements kills the pathogen directly by forming membrane attack complexes or indirectly by attracting phagocytic cells. Now, have a quick recap. At this topic, we have covered, or in this revision class, we have covered antigen, any chemical substance that evokes an immune response is called an antigen. Cell mediated immunity or CMI is the type of immunity where T cells directly attack pathogens that enter the host body or transplanted organ, and they also protect the body from its own cells that have turned cancerous. Humoral immunity, this is a triggered this is triggered by defensive proteins called antibodies which are produced by B cells. Antibodies circulate in the lymph, and the antigen antibody complex, an antigen binds with its corresponding antibody to form an antigen antibody complex to develop the immune response. An antibody has two identical binding sites called paratops for an antigen, and a paratop can bind to only one epitope, like a lock and key theory. The formation of an antigen antibody complex follows a lock and key mechanism, and these two types of immunities, cell mediated immunity and humoral immunities, are the type of coming under the category of acquired immunity. This is the writing assignment that you can complete to practice the topic antigen antibody complex. Describe the formation of the antigen antibody complex with the help of a diagram. Maybe you have already completed that because I have already given that exercise in earlier class. Here in this you can explain about this diagram and antigen antibody complex formation. Now try to understand today's topic with the help of some videos which make you clear about these facts in a proper manner. Now in this media presentation, we will see the action of B cells and T cells.
Action of B cells and T cells. How does our immune system protect us against infections and diseases? It does so with the help of special types of white blood cells. Macrophages, T cells and B cells. These cells identify certain proteins or polysaccharides called antigens on the surface of pathogens. When a macrophage identifies the antigen on a pathogen, it engulfs and breaks down the pathogen. Parts of the pathogen's cell wall appear on the surface of the macrophage. This signals other lymphocytes that a pathogen has entered the body. Once the T and B cells get this signal, they start to act on the pathogen. Let's see their mechanism of action. There are two types of T cells, T helper cells and T killer cells. The T helper cells recognize the antigens present on the macrophages. They begin to secrete chemicals called lymphokines that stimulate the production of more T and B cells. The T killer cells directly bind to the antigens on the pathogen and kill it by releasing cell wall dissolving chemicals. As T cells kill the pathogen directly, this type of immunity is called cell-mediated immunity. When a T helper cell comes across an antigen, it activates the B cell. The activated B cell binds to the antigen. This binding triggers the B cell to divide and produce memory cells and plasma cells. Memory cells are stored in case of a later encounter with the same antigen. Plasma cells respond to the antigen by producing antibodies. The antibodies bind specifically to the antigens, forming an antigen-antibody complex. The antibodies then deactivate or destroy the pathogens. As antibodies are released into the bloodstream to act on the pathogens, this type of immunity is called humoral immunity. Here is a summary of the immune response by T cells and B cells.
Now I think it is clear from this summary chart that our immune system produces two types of acquired immunity, cell-mediated CMI and humoral immunity that is developed by B and T cells. Cell-mediated immunity basically where the main role is played by T helper cells, and we have already discussed and explained that flowchart in the previous presentation. And in humoral immunity, main role is played by B cells by developing two different types of cells, plasma cells and memory cells. Plasma cells forms the antibodies and memory cells helps to memorize the structure of that particular type of pathogen in second time of encounter, which can develop the immunity at a faster rate. Now we are coming to this media or 3D where you will see particularly about how does our humoral immunity acts.
Humoral immune response. Humoral immune responses are immunological responses that are mediated by antibodies. During these responses, the immune system will trigger specific B cells to proliferate and secrete large amounts of specific antibodies to combat the pathogens. Let's take a more in-depth look at the entire process of the humoral immune response. An immune response begins once an antigen enters the body. The immune system will trigger specific B cells whose purpose is to produce large amounts of certain antibodies. The humoral immune response will begin with an activation phase. During the activation phase, a macrophage, one of the cells of the immune system, will engulf the invading antigen through phagocytosis. Inside the macrophage, a new vesicle called a phagosome will form. The phagosome will fuse with a lysosome. Inside the lysosome, digestive enzymes will break down the antigen particles, which will combine with class II major histocompatibility complex MHC proteins and move to the macrophage's outer surface. Macrophages with antigen fragments on the outer surface are referred to as antigen-presenting cells. Another cell, the T helper cell or Th, will recognize the antigen particles on the macrophage and then become activated. The receptors on the T cell will bind to the class II MHC protein and the antigen particles present inside it. After binding, the macrophage will release the chemical cytokine interleukin 1, which will stimulate T helper cells, causing them to release interleukin 2. The activated T cells will divide, meaning there will be an increase in numbers. The activated T helper cells will stimulate B cells, which will differentiate into plasma cells. The plasma cells will produce antibodies in large numbers, which will then flood the bodily fluids. The newly produced antibodies will bind to antigens and destroy them. Summary: the humoral immune response is an immunological response that is mediated by antibodies through T helper cells and B cells. Interleukins secreted by T helper cells will stimulate B cells. B cells will differentiate into plasma cells that produce large amounts of antibodies for combating the pathogen.
I think now it is clear about the humoral immunity, and now come to know about cell mediated immunity with the help of this medium.
Cell-mediated immune response. In another session, we described mechanisms of humoral immunity. In this session, we will discuss how T cells recognize and respond to foreign antigens. In the humoral immune response, B cells are crucial. In the cell-mediated immunity, T cells are important. Cell-mediated immunity is an immune response that does not involve antibodies, but involves the activation of macrophages, natural killer cells, antigen-specific cytotoxic T lymphocytes, and the release of various cytokines in response to an antigen. When an infection sets in, a virus will enter the body and will inject its genetic material into a host cell. The genetic material of the virus will produce multiple copies of virus inside the host cell. The protein recycling enzymes, such as lysosomes, will process the antigen and make them into fragments. The fragments of the antigen will combine with class I major histocompatibility complex, MHC proteins, and move to the outer surface of the host cell. Macrophages with antigen fragments on the outer surfaces are referred to as antigen-presenting cells. Another cell, the T helper cell or Th, will recognize the antigen particles on the macrophage and then become activated. The receptors on the Th cell will bind to the MHC protein and the antigen particles present inside it. After binding, the host cell will release the chemical cytokine interleukin 1, which will stimulate T helper cells, causing them to release interleukin 2. The release of interleukin 2 will stimulate the cytotoxic T cells. Cytotoxic T cells will proliferate and move around looking for other infected cells. When an infected cell that has the antigen along with the class 1 MHC receptor is found, the cytotoxic T cell receptors bind to the MHC proteins. After binding, the cytotoxic T cells will release pore-forming molecules called perforin. The perforin molecules will bore holes into the infected cell membrane and destroying the virus within the cell as well. Summary: the cell-mediated immune response is an immunologic response that is mediated by cytotoxic T cells. Interleukins secreted by T helper cells will stimulate cytotoxic T cells. Cytotoxic T cells will recognize infected cells and release pore-forming molecules called perforin to destroy the infected cell.
Now I think it is clear to all of you about the cell-mediated immunity also. And now come to watch this video where we will learn about the vaccination, which is also a type of technology by which we are inducing the immunity.
Types of vaccines. When pathogens such as bacteria or viruses enter the body, white blood cells known as lymphocytes or as B cells recognize them as antigens. In a process known as immune response, these lymphocytes will respond by producing specific antibodies to combat the antigens. The antibodies will destroy the antigens and also remain in the body to fight future infections. To successfully do this, a vaccine must mimic the pathogen or antigen. When vaccinated, the immune system will respond by producing antibodies for that particular antigen. This is the primary immune response. Along with the antibodies, memory T cells are also produced and are then stored in the bone marrow. These memory T cells recognize the antigen if they invade again and produce antibodies to quickly combat them. Thus, vaccines prepare the immune system to respond quickly and effectively should the body re-encounter the pathogen in the future. The main types of vaccines: live attenuated, inactivated, subunit, and toxoid. Live attenuated vaccines are prepared in a laboratory using the virus or bacteria in a weakened state, still alive and able to reproduce but unable to cause serious illness. Its mere presence is enough to cause the immune system to produce antibodies. Examples of these live attenuated vaccines are those which combat against tuberculosis, rabies, and smallpox. Inactivated vaccines are prepared from the dead pieces of disease-causing microorganisms. Because the antigens are dead, the strength of these vaccines tend to wear off over time. Since these vaccines have no long-lasting effect, Multiple doses are usually necessary to provide the best protection. Examples of inactivated vaccines include hepatitis A and B, poliovirus, and cholera. Subunit vaccines are prepared using only the part of the pathogen that triggers an immune response, such as DNA. Vaccines against hepatitis B and influenza are subunit vaccines. Toxoid vaccines are prepared from the inactivated toxin materials secreted by bacteria. These vaccines are effective on diseases in which the secreted toxins are the main cause of the illness. Tetanus and diphtheria vaccines follow into this category. Summary: vaccines are preparations that mimic pathogens and initiate the production of antibodies as a means of combating them. The main types of vaccines are live attenuated, inactivated, subunit, and toxoid.
Now I think it is clear to all of you about these important topics which are relevant in today's corona crisis also. Uh, these are the facts that a particular uh, medical professional should know about humoral immunity as well as CMI immunity to produce a particular type of vaccine, and in that way I have discussed with you today in today's revision class once again the important topic of the human health and diseases, that is acquired immunity, which help us to acquire the immunity against any type of pathogen in case of any infectious disease, and how our body will overcome that situation. That is the role played by the acquired immunity with the help of B cells and T cells. Now I'm closing our today's class is over. If there will be any doubt in this chapter, you can share with me in the class group so that I can take these topics also before your test. Now all the best for your test on Monday. Have a nice day.